Okay, welcome to Transcription Translation, the final lecture of the semester with my gecko dragon, who is the father of all the baby geckos I have. So, let's just get right into what transcription and translation are, right? So, transcription and translation refers to the central dogma of biology. Basically, what we're saying is that a constant rule in biology is that we go from DNA or gene to protein. We're just going to have to deal with that random vertical line or horizontal line that I hate so much. So, the gene is the DNA. And in order to express the gene, we convert it to protein. So the central dogma of biology is gene or DNA to protein. And transcription translation is all about how we accomplish that. So transcription is the process of converting the DNA gene into the RNA messenger. So we have a DNA gene, and it contains the information that needs to be processed there. It has all the information for the protein we're trying to build. So we're going to convert that gene into a sequence of RNA, right? So this is transcription. If you think about transcription, right, when you transcribe something, you are copying it, right? So you're just copying it, you're using the same language. Well, here we're copying, we're using the same language, nucleotides. So we are using nucleic acids. We're copying a DNA gene into an RNA messenger. You might remember this from way back as messenger RNA. And its whole job is to take the genetic information from the nucleus to the ribosome, where that genetic information will be used to construct a protein. Translation, however, is where we change languages, right? So in translation, we have our nucleic acid language in the form of RNA, the mRNA we produced in transcription, and now we need to translate it to a different language, and that language is protein. In other words, we're going from nucleic acids to amino acids. We are changing the basic language that we are using. So that is translation. So transcription from the gene, the DNA gene, to an RNA messenger that can leave the nucleus. That's transcription. And translation, taking the nucleic acid messenger RNA and translating it to an amino acid protein. So why do we go through all of this? The whole point is to construct proteins. And if you'll recall, way back when we talked about macromolecules, proteins serve a lot of important roles. They're enzymes, right? They're hormones. They're local messengers. They can be released by one cell to tell another cell what to do. They are antibodies, right? A huge component of your immune system is proteins. So proteins have a large number of different functions. that make them incredibly important to the biological system. Heck, just enzymes alone, right over here, govern when a gene is transcribed during fetal development. So if you have a developing fetus, right, 1 cell, 2 cells, 4 cells, eventually you're going to get to the point where you're going to start activating certain genes at certain times, and it's going to be the enzymes that really tell the cell what to activate when. So proteins govern basically everything. So what's the role of RNA in all of this? Well, as we said, you've got your cell right here, and it's got its nucleus, and its nucleus is full of DNA packed in there tightly as chromatin, right? And then we have our ribosomes elsewhere. And remember, the ribosomes are the site of protein synthesis. So ribosomes are not in the nucleus. Man, I hate those horizontal lines, but we got to go with them. So how do we get our genetic information in the genes out to the ribosomes? Well, we take one gene and we convert it to an RNA messenger that can then leave the nucleus. So here's a little gene that's available, convert it, and then it's able to leave the nucleus and eventually meet up with the ribosome. And then we can use that RNA to make the protein. There's some amino acids. So the role of RNA is to carry the information from the gene out of the nucleus, out to the ribosomes where synthesis occurs, because the gene itself cannot escape to the cytoplasm. All that DNA is too tightly packed in the nucleus. It's the role of the messenger RNA to take the information from a single gene out to a ribosome in order for synthesis of the protein to occur. Ribosomes themselves are primarily made of RNA. So we have messenger RNA that's carrying that information about the gene. You've also got rRNA, which is ribosomal RNA. And then you've got amino acids that need to be taken over to a ribosome, right? So what carries those amino acids to a ribosome? Well, what do you know? Oops. I found a new function that I'm not particularly happy about. Okay. There we go. Got out of it. So how do you carry that little, oh geez, man, clicking around. Gonna have to do some edits. How do you carry this little amino acid over to the ribosome? Well, you have yet another piece of RNA called transfer RNA. And transfer RNA's whole job is to take amino acids required for making the protein over to the ribosome where the protein is being made. Okay, we're getting a little repetitive. The instructions for every protein is encoded in the genes, right? And the genes are DNA. So, the genes are DNA, and that DNA gene is the instructions for the protein. So transcription is all about taking the DNA instructions and producing an RNA transcript. So the whole goal is to make a transcript of the gene, which is in the form of mRNA. We're looking for the mRNA transcript. So we copy the gene into RNA. This is termed DNA transcription. And of course, always remember that the whole point is to make an RNA copy. We're building mRNA. And it's that messenger RNA that can leave the nucleus and go to the ribosomes. So remember, we have all those enzymes in DNA replication, right? We've got in DNA replication, we've got topoisomerase, we've got helicase, we've got the single-stranded binding proteins, we've got the polymerase III, we've got primase, we've got, we've got primase, and we've got ligase, right? Those are all the enzymes operating together to replicate DNA. We've got 1, 2, 3, 4, 5, 6, and I'm missing something because there's a 7th. So we got topoisomerase, helicase, single-stranded binding proteins, primase, polymerase III, polymerase I. So, we've got 7 different proteins required for DNA replication. So now we're trying to take a gene and convert it from DNA to RNA. How complicated is that going to be? We've got transcription factors present in eukaryotes only. And we've got RNA polymerase II. Let's see how many more we have. Oh, that's it. That's it, folks. It's just transcription factors and RNA polymerase II. RNA polymerase II is the home shopping network version of an enzyme, man. It's, the commercial for it would be like, all right, folks, we've got some RNA to make, all right? Now, in DNA replication, we needed those 7 enzymes, folks. But now, today, I'm giving you RNA polymerase II. All right? RNA polymerase II is an amazing protein, my friends. All right? It takes all the functions of the 7 enzymes in DNA replication and does them itself. That's right. It's topoisomerase. It's helicase. It's primase, polymerase III, polymerase I, ligase. It does it all itself, my friends. That is 7 in 1. Call now, right now, to get RNA polymerase II for all your transcription needs. And if you call right now, we're going to double that RNA polymerase II and give you 2 RNA polymerase II for the low, low price of $19.99. Call now. Supplies are limited. Anyway, so RNA polymerase II is pretty amazing because it can do all these important functions on its own. So let's talk about all those super cool functions. First off, it's got to bind to the DNA at the correct site. Woo! Has to bind to the DNA at the correct site. What do I mean by that? I mean, it has to be at the beginning of the gene. Then it has to unwind the DNA to expose the template that it's going to use to build the mRNA off of. Right? In DNA replication, both the 5' to 3' and the 3' to 5' strands were templates for building DNA. However, in RNA transcription, only one is going to be the template. So we unwind the DNA, we expose the template, and as we do this, we just build the mRNA transcript off of the exposed template. So we're just going to start building, right? So here's your DNA. Polymerase III rolls up and starts to unwind it. So we unwind the DNA, get that unwound spot, and it's going to wind it back up behind it. And as it moves through there, it's going to be actively building the new transcript. So it reads the DNA and builds the complementary RNA off of that. And it builds that RNA as it goes along. And then, of course, as it passes through, it rewinds the DNA into its double helix. So how do we find the beginning of the gene? How do we find it? Well, there is a sequence, a DNA sequence right ahead of the gene called the promoter. We often refer to it as the TATA box. The TATA box is a TATA, thymine, adenine sequence that precedes promoters, whatever you might have, right? There's the TATA box. And so we also have a cat box, right? So the TATA box and the cat box are both sequences ahead of the gene that identify the beginning of the gene. So transcription factors are going to bind at the promoter, and these transcription factors, when they bind at the promoter, allow polymerase to bind more tightly. So here's some transcription factors. We'll just use some abstract little circles. Looks good, right? And it aids polymerase II binding. RNA polymerase II is aided in binding to the promoter via these transcription factors that bind to the TATA box or the cat box or the different kinds of boxes. So RNA polymerase II binds at the promoter sequence, and then it will move along the gene. It will follow the template strand and just move along until it's done transcribing. It binds at the promoter, and then it immediately begins laying down new RNA nucleotides to build the mRNA. So the template strand in transcription is the 3' to 5' strand. So that means if it's the 3' to 5' DNA strand, what direction is RNA polymerase II synthesizing in? What's anti-parallel to 3' to 5'? Yeah, that's right. Just like with DNA polymerase III, RNA poly II RNA poly 2 has to move in the 5' to 3' direction. Has to move 5' to 3'. So the template strand is the strand that's anti-parallel to that. So elongation is pretty simple. This is where we actually do our RNA synthesis, right? So here's our little Tata box. And we've got our gene. And then we've got polymerase II rolling along here. So here's my polymerase II. And then we've got the particular nucleotides on there. Let's just throw down some random ones. A, T, G, C, C, G, A, U, oh, not U, T, A, T, C, G. There we go. Some random nucleotides. And so we're going to build in the 5' to 3' direction. So if this is the 3' side of DNA, polymerase is going to be building the 5' nucleotides. And it's going to start moving along here. And as it moves, it plops down a nucleotide. So there we go. It'll plop down the complementary pair. Pops down a U. And then it moves a little bit further, right? And it plops down the next one. Right? And it continues elongating in this way. So at this point, and because it continues elongating with the first one here being the first one put down, you're going to have basically the RNA transcript reading like this, right? So here's the transcript we're making, and it's just being built like that. So it's just built along like that. So we're base pairing. The important thing to remember here, right here, is that we are base pairing from DNA to RNA. So remembering your base pairing rules, if there's an A in the DNA, like right here, the RNA complementary base pair is a U. Very important to remember. So elongation is just the fancy term for continuing the RNA transcript, making it longer as the RNA polymerase III travels down the template strand. And it will continue moving, elongating from the 3' end of the polymerase. So, right, we saw here's the 5'. So then we have 5', 3'. Right? So we're just going to keep adding in nucleotides here. All right. So how do we know when to stop, when to terminate transcription, right? Because we're only trying to transcribe one gene. How do we know when the gene is done, right? We know that a promoter, like the Tata box, tells us where to start the gene. How do we know when to stop the gene? Well, for termination in bacteria, they have the DNA terminator sequence. So basically what happens is RNA poly 2 travels along the gene here, right? Here's our little primer. And it will travel along until it reaches a specific sequence of nucleotides called the termination sequence. And when it hits this spot, right, here's our polymerase III, it hits this spot and it disassociates. It disconnects from there and it releases the mRNA that it just built. Pretty simple. A beautiful little terminator sequence. When RNA polymerase II reaches the terminator, it detaches from the gene and it releases the mRNA, and now you've got mRNA ready to seek out a ribosome. Pretty simple. With eukaryotes, it gets a little wonky, right? We don't have a terminator sequence. We can't do a nice, beautiful Arnold Schwarzenegger Austrian voice. The bacteria have a terminator sequence, and when the polymerase reaches the terminator, it is terminated. We disassociate it. Eukaryotes don't have a terminator. Oh, no terminator. How am I possibly going to make an Arnold Schwarzenegger reference? Oh, what's this? The polydenylation signal? Oh, so in lieu of a terminator, the polydenylation signal is produced on the mRNA transcript. Right? So here's our mRNA transcript being built. And when we reach a spot that gives us A, U, A, right? We know it's on the mRNA because there's a U. U is specific, it's uracil, and it's specific to RNA. So this sequence is my polyadenylation sequence. And this is what causes a cut to be made in the transcript. And it releases the growing transcript from the polymerase and the polydenylation signal. Now, here's the thing. This transcript is not immediately ready to leave the nucleus and go be turned into protein. What I love about the polydenylation signal is over here I'm going, oh, the terminator. The terminator terminates transcription. Well, down here we have the polyadenylation signal. And basically, we know we have to stop when we get the Arnold Schwarzenegger manly scream. Right? That's a scream from Conan the Barbarian. When they chop him in the arm or something, he'll go, and so they're cutting him with the sword. That's a downstream cut, right? This is a metaphor that is really reaching because I just like making Arnold Schwarzenegger noises poorly. Okay, so our transcript that is produced when we cut it from the polyadenylation signal, right? This is called pre-mRNA. So here's my little sequence that was cut off. Here's my polyadenylation signal. My right? I've cut it right here. So I've released the transcript, and this transcript is called pre-mRNA. Right? So in bacteria, it's ready to go. As soon as we hit that terminator and we release the transcript from poly 2, it's just ready. It can go straight to the ribosomes. But in eukaryotes, it's not ready. Pre-mRNA is not sent to the ribosomes. It does not yet leave the nucleus. Instead, we have to do some processing. So here's a nice little picture of our RNA polymerase right here. And it's coming in and lifting the strand open, exposing my template. This is my template strand, right? And we can see that it's the 3' to 5' strand, such that as polymerase 3 is moving along it, it's moving in the 5' to 3' direction, anti-parallel. And it's just building, slapping down RNA nucleotides. So here's my newly made RNA. Whoa. I'm going to try and grip this differently. Keep pressing a button. Here's my newly made mRNA there. And it continues being produced from the 3' end, right? So here we can see, because we're making something anti-parallel, our template's 3'. So our transcript is 5'. And we add new nucleotides down on this 3' end. So we're growing basically from the front. So if we continued along, say this was the very beginning, this would be the first complementary nucleotide that we slapped down, if that was the beginning there. And we just continue moving. So the first nucleotide transcribed is sort of the tail end of our transcript. So just we're putting down new nucleotides from the 3' end. We're elongating from the 3' end. This is a good movie.
Transcription is the process by which RNA is assembled from a DNA template. The two strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites.
Okay, so there's the coding region, right? And then ahead is the promoter site, and then they showed the termination site.
A molecule of RNA polymerase binds to the promoter
binds
site.
to the promoter and starts.
It moves along the DNA, separating the two strands of the double helix.
There you go.
Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base.
Starts building the RNA transcript.
In the synthesis of RNA, uracil is the nucleotide complementary to adenine.
Remember that if the template strand has an A, the transcript has a U. We just build that transcript.
This process ceases when RNA polymerase reaches the termination site.
This is bacterial, so
the DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
There we go. So if this weren't bacterial, we'd actually have the polyadenylation signal in that RNA transcript. And then we'd have to cut the polyadenylation signal from the RNA transcript in order to release the transcript and have it ready to be processed. So now we're going to do RNA processing. This involves adding two portions, one on each end. We add the cap and the tail. You'll never guess to what end the 5' cap is added. Huh? Oh, yes, the 5' end. So here's our transcript. And here's our 3' end and our 5' end. And we add a 5' cap, which is just a series of RNA nucleotides designed to withstand the immune system of the cell. So it's just like a little bit here that basically tells the cell, hey man, this genetic information is cool. The reason this exists here is because there are viruses whose genome is RNA. And so they'll inject RNA into the cell, and the cell will have to, try and recognize whether or not the RNA getting produced is its own or viral. So basically, most eukaryotic cells chop up any RNA that's present in the cell. And the only thing that prevents chopping is a 5' cap and a poly A tail. Anyone want to guess what the nucleotide makeup of a poly A tail is? Huh? Oh, adenine. Why, you guys are geniuses. So the 5' cap is sort of like an attempt to identify it. Hey man, I'm cool, I'm you. That's great, but it's still going to start getting chopped up, right? The enzymes that defend against RNA, against viral RNA, still going to start chopping. So the poly A tail is great because as we get some chops in here, we don't lose things. So we get a chop. And we sort of chop the poly A tail. One of the advantages of this is actually that how much poly A tail there is governs how long the mRNA is being used by the cell. How long the genetic product, how long we're making the product of the gene, right? The longer the poly A tail, the longer the mRNA sticks around in the cytoplasm being translated into protein. Because once we lose tail, chop, we chop that RNA up. Chop it, and it's unusable. So once we lose that poly A tail, we can't protect the mRNA. This actually is great because you don't necessarily want to constantly produce the product of a gene. So a poly A tail can be of different lengths, and that can have a built-in timer of how long the mRNA lasts and how long you're making that transcript product. Right? That's pretty cool. So the cap is like an identify friend thing, like, hey, pal, don't chop me up, bro. And the poly A tail is just like, yeah, I know you're going to chop me, but why don't you chop this stuff that isn't part of the gene first? So it protects the transcript from the cell's own defense against viruses. It also aids the transcript in binding to the ribosome. Okay, the next important thing is editing, right? That wasn't just all of RNA processing. It's not just the 5' cap and the poly A tail. We also have to do some editing, right? So if you look at DNA, here's the gene that produced our transcript. Portions of that gene will be what we often refer to in the science as non-coding regions. So maybe here is a stretch of DNA that's not involved in the protein, but is still part of the gene, right? It's a non-coding region in the gene. Sorry about those annoying horizontal lines. I went online to try and figure it out, and the best answer was delete PowerPoint and install an older version, which is I'm too lazy for it. So here we have non-coding regions. So this is our gene, which means when we make our transcript off of this gene, our transcript will also have non-coding regions. This is bad, okay, because a protein has to have a very specific structure. Has to be folded in just the right way, presented in just the right way. So we don't want all these non-coding regions there in our transcript to mess up the protein. So we're going to remove our non-coding regions. So we separate out our coding and non-coding regions into introns and exons. And then we remove the introns and splice together the exons. So this non-coding region on the transcript is called an intron. And we just cut out the introns, and these coding regions are known as exons. So we need to cut out the coding regions and splice together the exons so we have one long transcript that only has coding information. This is our coding transcript. In which we cut out the introns. And we spliced together the exons. So all our introns have been removed. So we cut out an intron and we splice together the exons. So how do we know where to cut? My gecko is still where he's supposed to be. Well, there are these sites that are called splice sites. So we have these little molecules called Snerps. I love that the actual scientific term is Snerp. snRNP is the actual acronym. But any biologist will call it a Snerp. So a Snerp binds to the introns. So let's get our transcript back up here with its introns and its exons. So here's an exon, here's an exon. I'm going to be lazy and leave it at this. Whoop. Woo. So we've got our red introns, or our red exons, and our little blue introns. And our Snerps are just going to bind to introns, and they're going to cause a curl. So our intron curls and binds up to that Snerp. Ugh, stupid horizontal line. Right? So we bind up to that Snerp, and then we can just cut out that curl and splice together the exons. So check it out. Here is a drawing. Here's one Snerp on one side of the intron, and here's the other Snerp on the other side of the intron. And we create this loop. And the Snerps pull it into a loop, and then we cut the loop, removing the intron and splicing together the exons to make a coding mRNA. So our finished product, right? Let's run through the list, right? We had our template DNA, which is 3' to 5' DNA, so that polymerase moved in the 5' to 3' direction. For some reason, that's the sweet spot for these enzymes that build nucleotide chains. All right? We built our transcript. This is a eukaryote. So here's our transcript growing out. And then we hit our polyadenylation signal. Oh my gosh. Keep pressing the button. Once we hit that polyadenylation signal, we snipped the transcript downstream of it, cut, and this was our pre-mRNA. Our pre-mRNA then had the introns removed and spliced together all the exons to make an exon-only transcript. And of course, we added the 5' cap and the poly A tail. Now that we have a transcript that is made only of exons, that has a 5' cap and a poly A tail, it is functional mRNA. It now fully contains the instructions for the proper protein, and it will be used to make the protein. And that, of course, is done in the cytoplasm. So it's finally time for our transcript to leave the nucleus and head to the cytoplasm. Hold on one second. It is doing a weird thing. I'm just going to cut this. Okay. All right. Now that that's cut, we're making the protein in the cytoplasm now. So transcription is complete. Bacterial transcription, so much easier, right? Poly 2 comes in, builds the transcript, elongating from the 3' side until it hits the terminator, releases it. The eukaryotes were more complicated because of that polyadenylation signal, requiring us to cut that transcript free and requiring us to process the transcript, removing the introns, splicing the exons, adding the 5' cap, and adding the poly A tail. Right? There was quite a bit of processing to be done in eukaryotes only. So now we're on, finally, to making a protein. So this is translation. Translation is where we go from one language to another, like you might have Spanish to English, or English to Japanese, whatever. Here, we're going nucleotide to amino acid. That's what we're translating. The players involved in translation are our functional mRNA, a ribosome, which is the site of protein synthesis, tRNA, which brings amino acids to the ribosomes so that we can construct the protein. And of course, since I mentioned them, the amino acids that the protein is built out of. So let's look a little bit more into the ribosome so that we can have some details on how this whole thing works, right? So obviously we've said it a million times by now, but the ribosome is the site of protein synthesis. So let's talk about the details of what that means. A ribosome has to have a spot where it can bind to and, quote, read the mRNA, and it has to have a spot where we can add amino acids. So here's my little ribosome, a large subunit and a small subunit. Here's my mRNA right here. And it binds to my mRNA. So it binds to the mRNA, and then as soon as it binds, a tRNA rolls in, carrying an amino acid. So here's my little tRNA. And it kind of looks like a little T. It looks like something like that. And it is carrying a little amino acid. So here we go. And here's an amino acid. So it comes over. And it comes over, it attaches to the ribosome, and deposits in its amino acid. So, transfer RNA. It carries the amino acids from the cytoplasm. We've got a lot of amino acids sort of loose in the cytoplasm that attach up to the tRNA, and then the tRNA is attracted to the ribosome and the mRNA, and it brings in the correct amino acid, which then gets added on to the growing chain. So how do we go from nucleotides, which come in 4 flavors in mRNA, that's A, adenine, G, guanine, C, cytosine, and U, uracil. There are just 4 of these nucleotides. But we need to be able to produce 20 common ones at least in an animal. So what do? Well, we arrange our nucleotides into codons. A codon is a sequence of 3 nucleotides. So we read the nucleotides on the transcript in sets of 3. Each set of 3 is called a codon. And each codon codes for an amino acid. And this gets us a lot of variability in how to code for amino acids. In fact, the 20 amino acids often have, for the most part, multiple codons coding for the same amino acid. So, one codon codes for one amino acid, and we have some very important codons to mention. A transcript always starts with an AUG because that is the start codon. That's where the whole thing gets going, right? AUG is the start. In fact, when the mRNA first arrives at the ribosome binds around the start codon. So here's my ribosome, and here's some mRNA, and the ribosome specifically binds to the start. It binds right to the start. Now, how do we know when to stop? Well, just like there's a start codon, there are stop codons. Those stop codons come in three flavors. We have UAA, we have UAG, and we have UGA. Any one of these three will cause protein synthesis to stop, and will release the growing protein, and that'll be the end. So this is something you're going to need to be intimately familiar with, because I guarantee you that this table is on the final. So you'll see they have first letter, second letter, third letter. Well, there are three nucleotides in a codon. So we know AUG is the start, right? AUG. So we look for the first letter A. We look for the second letter U. And we look for the third letter, right? So we go, here's the U. And here's the A. So we know we're in this box here. And we just look for which one is the G. So here's the G. And we can just follow the G down its row. So follow over to our box, and here's methionine, and it's green because it's the start. Yes, AUG codes for methionine. So you can have AUG multiple times in a transcript. As long as there's no stop, it doesn't matter if you have multiple starts. Only the first start matters. Okay, so let's talk about this whole process. And step one is mRNA arriving at the ribosome. And the first codon red is the start, because that's what the ribosome binds around. Right? So here's my ribosome, my large subunit, and here's my small subunit. And we bind to our start codon. I hate those graphical artifacts. There we go. A little bit of junk there. So we bind to that start. It's AUG. That codes for methionine. So, tRNA has its little methionine, right? There's a tRNA for every amino acid. This one's carrying methionine. Get our methionine on here. There we go. And it's going to arrive and bind here and release its methionine. And we'll get a methionine down, MET. So how does tRNA know to bind to the ribosome and to the mRNA? Well, mRNA has codons. Wouldn't it be really convenient if tRNA had something that could be complementary to an mRNA codon? Oh, maybe an anticodon? Yes, tRNA has anticodons. An anticodon is basically a complementary set of nucleotides, right? So here's my tRNA carrying methionine. Here's my little drawing. There we go. So we got methionine. We know that our mRNA has the AUG codon for methionine. So the tRNA anticodon is literally just the nucleotides that are able to base pair with the correct RNA codon. So remember, tRNA, so this is not a U, right? Or this is not a T, excuse me. This is a U, A, right? U to A to U, C to G. So the anticodon can base pair with the mRNA codon. So the anticodon is tRNA. Anticodon base pairs with the mRNA codon. Not too terrible. But this is how we get the right amino acid to the right spot on the ribosome, because tRNA base pairs with codons. Each tRNA has its own anticodon that base pairs with an RNA codon. So if we look at that table over here, right, we can look at any one of these, right? Say we're looking at UCU right here. UCU is the mRNA codon that corresponds to serine. The tRNA anticodon that would allow serine to be brought to the ribosome would be AGA. Not terrible. So here's another little drawing of our codons and anticodons. Here's the start codon on the mRNA, AUG, and here's the tRNA anticodon, UAC, so that we can bring methionine over. Again, here's another pretty drawing showing basically the same thing. All right. So we've started off. The ribosome binds to the start codon. It puts itself together around the start codon. Then tRNA with the UAC anticodon binds to the AUG codon and brings methionine. What now? How do we go from methionine to a polypeptide? A polypeptide being a chain of amino acids. So here's 1, 2, 3, 4, 5, right? 6, 7, right? With that first one down being methionine. How do we get from methionine to a chain? Well, as always, we start with that start. The anticodon UAC binds to the codon and brings methionine. What happens next? Well, that's all based on the ribosome. The ribosome actually has 3 binding sites that associate with mRNA. And they are called the A P E sites. So, these are 3 sites. Each site corresponds to a codon. So when the ribosome assembles, it assembles around the AUG at the P site. And then we have the next codon and the next codon. Right? And so we have the A. I like to call the A site the awaiting. Right? We're awaiting adding our amino acid. We're awaiting. The P site is the processing site. Where we actually add the amino acid on. And then the E site is my exit site, where the tRNA leaves. So, at the awaiting site, the tRNA arrives at the proper codon. At the processing site, the amino acid it's carrying is added to the chain. And then at the exit site, the tRNA leaves. So, we know how this works. We plop down our methionine. So here comes our tRNA with its anticodon, UAC. I'm not going to, whatever, the drawing sucks. But it drops its methionine down. At the same time, the tRNA with the anticodon for this guy at the awaiting site arrives. And he's chilling, waiting to add his amino acid. Why don't we just find out? Let's go back and find out which amino acid corresponds to CAC. We'll just look at that table again. So, CAC, our first letter, C. Oh, man, I hate the little button on this pen. Our first letter is C. Our second letter is A, meaning we are in this box. And our third letter is C. Follow that over, CAC, and that is histidine, H-I-S. You do not have to know the names of each of these amino acids by heart. So, boom, boom, boom. Okay, drawing is getting a little crazy because of all those stupid horizontal lines. But at the awaiting site, we have, oh, that cleared up a little at least, our tRNA with its anticodon that fits to the mRNA codon and the amino acid it's carrying, histidine. So we process that methionine and begin our chain. And then we chunk a chunk forward. Oops. Well, starting to look like it'll be easier to just erase this and start over. Okay, so we chunk forward. Ribosome. mRNA. We've moved forward one. So our AUG right here, our CAC, our GAG, and our UAA. Right? So again, we have our EPA. So now, again, we have the little tRNA with the correct anticodon here, C, U, C, U. Hangs out in the awaiting site, bringing along its little friend, CAC. It's GAG. A problem that I just had that you might have, you need to be aware of, is when you mistake the anticodon and the codon in your head. I was thinking CAC because that's the mRNA that's the tRNA anticodon, but that's a mistake. I have to go by the mRNA codon, which is GAG. GAG, which gives us our, I think, glutamic acid. So here's my GLU. Over here, we've got our little methionine already added. And now we have our little tRNA at the processing site. And it's just going to chunk on its histidine. Doop. Right, and now we've got two amino acids, methionine and histidine. Over here, our empty tRNA, right, it lost its methionine. It is released back into the cytoplasm where it will pick up a new methionine and be ready. Man, these tRNAs do not look right. Anyway, that's how it goes. We just continue chunking through, adding stuff on until we get to a stop codon. So A site awaiting The tRNA is there awaiting adding its amino acid to the chain. It's a little pre-bind. The P site is our processing, where we actually add the amino acid to the chain. And the E site is exiting, where the tRNA actually gets to exit now. It can release from the mRNA and go find a new one of its nucleotides that it carries. We continue and continue until we hit a stop. So, not too terrible, I hope. Here's a little picture of the EPA sites, awaiting, processing, exiting. So, this drawing indicates to me that it's right at the beginning, because we don't have anything in the A site or the E site. So the ribosome has assembled around the mRNA at the start codon. Anyway, got another little video. Minute 30.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. An aminoacyl tRNA bound to elongation factor TU, EFTU for short, enters the free A site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. Elongation factor TU hydrolyzes its bound GTP and dissociates. If the tRNA is correctly matched and remains bound...
There is extra stuff in this, by the way. EFU, GTP, this is like extra stuff you don't need to know.
To extend a growing...
You know what? Let's just mute this sucker. So we've got our A, P, and E sites. The ribosome has assembled around an AUG. Then here comes a tRNA, but as they mentioned, it has the wrong anticodon. So when it arrives at the A site, it tries to bond, but it's not compatible. It doesn't have the complementary base pairs. So it's rejected, and that'll happen multiple times until the right anticodon arrives, and we can get that hydrogen bond. Right? And so then we move and we click forward. Just need saying a bunch of stuff. And then we can exit stuff. So this animation is a little more correct, but it makes a little less, I don't know, brainy sense to me. So anyway, just an attempt to show you how we continue elongating from there. So at the P site, we actually connect the amino acids together in order to make our chain. So we continue on, and how do we finally stop? Once we hit that stop codon, what happens? What happens at the stop codon? So there is no complementary tRNA to a stop codon. There's no tRNA carrying an anticodon that is relevant. Instead, when a stop codon arrives at the A site, a release factor binds, and we have water resulting in hydrolysis of the bonds, releasing the amino acid chain. So the release factor contains water, binds to the stop codon at the A site, and we hydrolyze our growing chain, right? So here's our ribosome. Here's, some random stuff. And here's a stop codon, right? And so this is the A, this is the P, this is the E. So release factor is going to bind here at the A site and donate water, causing hydrolysis. So here's our little growing amino acid chain. And our release factor arrives. Let's make it blue because it carries water. It arrives and binds and says, hey, you, break off. We hydrolyze the bonds and we release our new amino acid chain out into the cytoplasm or the rough ER or whatever. And from there, it can be it can go through its levels of protein structure. Remember, a chain is just the first level of protein structure. It's primary structure, a polypeptide chain. Secondary, we have our little helixes, right? And we have our beta pleated sheets. Right? And then after that, you get the big compact ribbon for tertiary, and sometimes you get quaternary where you have multiple tertiaries together. Anyway, that's besides the point. What we were saying is when a stop codon hits the A site, a release factor binds. There is no tRNA anticodon. There is no tRNA anticodon involved. A release factor binds, uses water to hydrolyze the bond and release the amino acid chain. So, sometimes you need to carry a polypeptide to its ultimate location. And I always thought this was kind of cool because you'd think the polypeptide would migrate to its ultimate location after release factor chunks it off and releases it. But no, in fact, the polypeptide is migrated before translation finishes. So the polypeptide is moved before the end of translation. So the whole ribosome, tRNA, mRNA complex moves to the ultimate location as a whole, so that when release factor binds to the stop codon and breaks it free, it's already where it needs to be. I thought that's pretty cool. And the reason it's able to be taken to specific sites in the cell is because there is a signal peptide. So what we're saying is that you'll have your amino acid chain and some sequence of amino acids in there results in a signal. And that signal peptide tells the cell where to go, or tells the cell where to put this. So this is my signal peptide. It's literally just a specific sequence of amino acids that's a signal. And the whole chain, once that signal is exposed, is grabbed, ribosome, tRNA, growing chain, and all. It's grabbed by a signal recognition particle and taken to its ultimate location. So, as soon as we produce the signal peptide, a signal recognition particle binds and drags the whole complex, ribosome, tRNA, growing chain and all, to its ultimate location. I thought that was pretty cool. So the last thing we'll talk about are mutations in the genome. So what is a mutation? A mutation is an alteration of DNA, specifically alterations of the base pairs, right? So if I have A, C, G, A, T, T, I always remember that there's base pairs, and this is DNA, so I'm using thymine, G, C, T, A. So here's maybe how it's supposed to go. And then a mutation occurs, and we alter at least one base pair. And instead of being a GC pair, it becomes an AT. That's a mutation, an alteration in the DNA. Specifically, this is called a point mutation. A point mutation is where you alter one pair, one base pair in the DNA. One base pair. Maybe it goes from GC to AT, CG to GC, you know, any of the different ways. A point mutation alters one base pair. There are 2 or 3 basic ways we can classify these. A lot of point mutations are what we call silent mutations. So what this means is that even though there's a mutation, a change in the base pairs, it does not result in a change in gene expression. And let's take a look at why. So, say we've got our genes, and they are A, T, G, C, A. So we've got, there, 2. 2 codons there. Always got to remember, this is the DNA, so I'm going to put down my complementary strands, and I'm going to use my thymine because I'm still in DNA. And we have a point mutation. So to determine if it's a silent mutation, we got to think about what the mRNA produced off of this is. So this is our 5' to 3', and this is our 3' to 5'. Meaning this right here is our template. So TAC produces AUG, start. And then GTA is going to produce CAU. So that is CAU. Histidine. Now, say we have a point mutation. Oh no! A mutation occurs. What could possibly what could we possibly do? And we lose this A and T. We get a mutation, and instead of A and T, right? Mutation. We get G and C. So now on our template, instead of an A, there's a G. So instead of CAU, we're going to have CAC. So C, A, C. Well, what do you know? Histidine. So this point mutation produced no change in expression. So it is a silent mutation. A silent mutation because it did not produce a change. no change in expression. A missense mutation is where you get a different amino acid. So we actually do change. So if we go back to our little drawing over here, what if instead we end up with a GTA was our template. Let's get to our colors. So our template was GTA originally. And say there's a mutation, right? The mRNA produced from that was CAU. So original template. There's our original template DNA. Here's our original codon, which is always referenced. If I say the word codon, I'm talking about mRNA. CAU. That gets us histidine. And say instead of a point mutation that doesn't change it away from histidine, we get a point mutation and suddenly it becomes G, our template mutates, and we get GTT, producing an mRNA that is CAA, and CAA is right here, producing GLN. Maybe, I don't remember what that one is. So, GLN. So now it's missense. It's missense because we have changed the amino acid produced. We've now got a change in expression. Now, just because it alters the amino acid expressed does not mean that it's a negative mutation. A lot of missense mutations don't do anything. In order for a missense mutation to drastically alter protein function, it has to occur, say, at the active site of an enzyme, right? Remember that active site has to have a very specific shape to fit its substrate. So if you get a missense and it messes up the active site's shape, then you can screw up the protein. If you get a missense in a vital structural area of the protein, you can mess it up. But for the most part, statistically speaking, your missense mutations, even though they're altering the amino acid produced, are not going to produce negative effects. In other words, point mutations are mostly harmless. The entry on humans on Earth in the Hitchhiker's Guide to the Galaxy. Mostly harmless. Like I said, unless you hit the active site or an extremely important structural spot. And then if you hit that, it becomes a nonsense mutation. A nonsense mutation means we totally screw up the transcript. So there are different ways to hit a nonsense mutation. One of the drastic ways you can hit a nonsense mutation, let's go back to our little table here. So say we have our original template. All right. Here's our original template. TAC A G A C A and then A T T. right? Let's build our transcript off of this. We get our mRNA that goes AUG, UUC, UGU, UAA. Right? There's start methionine. UUC is phenylalanine, I think. UGU is going to be cytidine, and UAA is a stop. Let's put a nonsense mutation in here, a drastic nonsense mutation. Ready? Our drastic nonsense mutation is where we have our template that has our A, T, T, and we get a mutation, a point mutation, and it drops it to ATG. And that gets us UAC. UAC is tyrosine, right? UAC, here's our U, here's our A, here's our C. Look at that. Due to a point mutation, we lost our stop codon. So the transcript was supposed to end, we're supposed to be done, but we lost the stop codon. So we can't bind a release factor. We can't release that sucker. You're just going to keep making it. So you're going to keep going past the original stop codon, and you're going to make something nonsense. So because that point mutation lost us the stop codon, we ended up with a nonsense mutation. So, if it hits a vital area in the active site, if it hits a vital structural area, or if it removes a stop codon, then boom, it's nonsense. So here's some point mutations. Here's our original sequence, right? We have the CAA, right? And then they mutate it to CAG. CAA coded for GLN. Well, CAG codes for GLN. So that's a silent mutation. Then they had the UCU right here. And it mutates to be CCU. And we go from threonine, right? Oh, CCU. We go from threonine to proline. So this is at the very least a missense mutation, not necessarily terrible. But then we have a UAU right here, which codes for tyrosine, and it gets mutated to a UAA, or the DNA that produced the UAU gets mutated to and produces a UAA, and we get a stop. And now we just have methionine, and boom. So we have silent mutation here. This first one is silent. The second one is a missense. And this third one is a nonsense. Finally, we'll talk about the reading frame mutations, which are a particularly drastic kind of mutation. So, a point mutation is where we mutate one base pair into another set of base pairs. A reading frame mutation has to do with what we learned in chromosomal inheritance when we talked about deletions. In a reading frame mutation, we have either an insertion of new base pairs or a deletion of old base pairs, original base pairs. So you can have an insertion that adds one or more base pairs, or a deletion that removes one or more base pairs. And this produces almost always some pretty drastic results. So here's a frame shift, right? We have, let's see, this is a terrible, they have the template DNA wrong, because if this was RNA, it'd be right, because that would be a AUG, but let's draw our own reading frame mutation real quick. So let's do an insertion. There we go. Okay. So we have our template DNA. TAC, AUG, GAC, and TAA. This is our template DNA. Right? Then we're going to make sure we write down our RNA, our codons. Easy. I always start with a start. And in an example like this, I always have an end, a stop at the end. And then we're going to have our UAC. You know what? That might be a stop codon. I'm not sure, though. CUG. Let's write down the amino acids we get. I know a couple of them offhand. Methionine, stop. And let's, of course, go to the table. You by now I'm hoping you see that you're going to use this table an awful lot, just a ton. So UAC, CUG. UAC, CUG, UAC. Oh, good. Tyrosine. So TYR, CUG, leucine. All right. We have now properly gotten our amino acid sequence from our original template DNA. Boom. Now we'll just put an insertion in. And this is where things get wacky. And this is why we call it a reading frame mutation. Because here's codon 1, codon 2, codon 3, codon 4. But now I'm about to insert a base pair. Suddenly I have C in there. And now let's look at the codons. So we still have the reading frame normal here, TAC, AUG, but now it reads CGA, CTA, and that A doesn't have anything going on there. So we can immediately see that we've lost the stop codon. Losing the stop codon always screws everything up. So we still have AUG. We still have our AUG. We still have our UAC. But now we're going to have CGA, CTA, and then just some random A. So no more stop. Our stop codon is gone. That right there screws the whole thing up. But the reason it's called a reading frame mutation is simply because when we put that mutation in there, we altered how we read the codons. We changed the codon sequence. We changed the reading frame. The frame is 3 nucleotides at a time. Adding in a base pair changed the frame. And deleting a base pair changes the frame just the same. So say we lose that. Now we've done a deletion. Get that. There we go. So we have TAC, AUG, ACT, AA. The deletion also changed how we read it. So these reading frame mutations almost always produce nonsense. And my gecko just oh no. You're trying to be a sneaky little gecko, aren't you? So this is almost always a nonsense producer. It's rare that a reading frame mutation won't just mess everything up. So this is a drawing that attempts to show a frame shift, but they get weird on this because, I mean, ATG is DNA, right? So that's not a start codon. That's DNA. And that would translate into a UAC, which is not M-methionine. Methionine is AUG. So, man, they messed this picture up. Screw you, picture. I did better. All right, mutagens. Mutagens are things that cause mutations. Not too terrible. A mutagen is anything that tends to cause mutations. It can be a physical substance. It can be a chemical substance, physics. When I say a physical substance, I mean something like energy, like gamma radiation is a mutagen. And then you can have chemicals like asbestos. So physical things like gamma radiation, chemical things like asbestos, these are things that cause mutations. Technically, anything that's chemical is physical, but a mutagen causes mutations, whether they're point mutations, deletions, insertions that make reading frame mutations. A mutagen causes mutations. The grand majority of mutations in your body are either harmless, which is, I would say, the grand majority. Then after that, you get mutations that are harmful, which your body will often destroy. And then you get, very rarely, the beneficial mutation. Everybody likes to think of evolution occurring through mutation, but evolution does not occur through mutation. Every single living thing has a large variety of different traits coded inside them. Look at domestic dogs. They're indistinguishable from wolves when you get into their genome. But look at the breadth of different traits we've managed to tease out of the wolf genome to get different domestic dogs. So any organism actually has vast amounts of different genetic information hiding in them. So mutations are not the driving force of evolution. So always useful to write down. So lastly, we repeat ourselves. The central dogma of biology is taking a gene, transcribing it, copying it into RNA, and translating it to protein. Gene to RNA to protein. That's the central dogma of biology. And that, my friends, is the very last lecture. So, parting is such sweet sorrow. Gecko says goodbye. Oh yeah, I guess all cellular life exhibits the central dogma.